Okay, so I think we will start with the last session of the conference and last session before the beautiful weekend ahead of us. And I'm really pleased to announce our presenter, Nick Goldman, who seems to be in Cambridge for a long time. Nick Goldman did his undergraduate in mathematics, so he started as a mathematician, actually moved into biological sciences. From there, he did it in Cambridge, left for a bit, came back to Cambridge again to the Department of Zoology, and graduated as a PhD in molecular genome evolution. Actually, that's where all the story started. And after that, after a while, he actually moved to European Bioinformatics Institute, which is a part of European Molecular Biology Laboratory, and is based, again, next to Cambridge, UK. So the focus of Nick and his group is to develop data analysis methods to study molecular sequence evolution, and then to exploit evolutionary information to draw inferences about phylogenetic history and genomic function. So they both work on research projects, and they both work on making the tools available for the scientific public so they can use it for their own research. So they are developing new evolutionary models and methods and use them then for comparative genomic studies. And it's really, really interesting and exciting that Nick Goldman had significant contribution in creating the field of DNA storage that I hope that we will hear about today, and actually to use DNA as a way to archive digital information, because as you know, we're getting more and more of information now, and we're getting the challenge how to store this and how to make it practical, how to have a storage that has high capacity and also that has low maintenance. And it appears that the DNA is suitable on all these parts. So I'm really looking forward for the lecture. And Nick Goldman.
Oh, thank you. Can you hear me okay? Can you hear okay? Okay, great. Thank you so much for the invitation to this meeting. This is my first visit to Lithuania. I'm really sorry it couldn't be a longer one, but the state of British politics at the moment means I want to be home tomorrow. I'm 55 years old and I'm going to demonstrate on the street in London, which I thought would never happen. Okay, so a couple of questions. So I think I kind of know the answer because I heard about what this meeting is. But how many people are currently students or graduate students? Could I just quick show? It's not a test, so lots and lots. That's what I expected. How many people in the audience are supervisors of graduate students? It's also not a test. I mean, yeah, okay, a few. All right, I just need to know quite how I'm going to pitch it. It works either way, but I just want to. That's really useful. And one more question. How many people know who James Joyce is? Is that so some? So it's kind of known. It's all right. It will be explained, but that's good to know how well known. So James, quick version is James Joyce was an Irish author, but you'll find out more later on about where we get there. So it's not the normal science research talk. Hope that's okay. I will talk about DNA storage. This comes about because of it, but I have a different story to tell and a moral at the end. And the moral gets adapted depending on the audience. So we're going to do the version that's more for students, but we can do them all. I'm going to tell you a story, and the story or the play is in 4 acts. And the first act took place in May and June of the year 2010. And I don't suppose scientifically any of you remember particularly what happened then, but it was this. This is Craig Venter and his team at the Craig Venter Institute. And they made a living bacterium they called Cynthia. So Cynthia in English is a woman's name, but also shares a sound, though not quite the spelling, with the beginning of the word synthetic. And this was a bacterium where they artificially created the DNA, the genome of the bacterium, and inserted an artificial genome based on a design from a living one into a living bacterium. And it lived and reproduced and is still alive somewhere in a laboratory. And this is a really cool story, obviously, that we understand enough about manipulating DNA and what's in a genome and how genes work that we can't create the whole living organism from scratch, but we can kind of rewrite the genome, which of course has always been described as the hard disk drive of life, the instructions for life. And humans can now make that. So there's some pictures. There's a little page from the publication which was in the journal Science, and another picture and a kind of schematic of the genome. And one of the things they did with the genome is they added bits that didn't have any biological or cellular function, but contained a message. And it doesn't show, is it that one? No. Is it? Got it. Thank you. And on this schematic, they're called watermarks. WM1, WM2, WM3, and they called them watermarks. And these are regions of the genome where they wrote some DNA into the genome that had no biological meaning, but had information in that they put. We'll come back to that in a little bit. But it depends on the audience. But it's helpful if I tell you what DNA is for anyone who doesn't know. So DNA we think of as being a living thing. It's in our genome, but it's just a chemical. It forms a polymer made of 4 molecules conveniently known by the letters A, C, G, and T, but representing 4 fairly small organic molecules. And those will join together in a chain that can be very stable, exceedingly long, millions of elements long, easily. And in the sense that if you choose, or if nature or biology chooses the sequence of the letters, it can contain information. So the most famous example of this, of course, is the protein coding parts of a living organism's genome, and that's probably the most famous because it's the bit where we understand the code best. And we know that if you read in 3 letters at a time blocks, that forms a code which can be translated to the building blocks of proteins that makes all the components of a living body. So on the right-hand side is little pictures of the molecule, and the famous double helix is in the middle, which is one of these chains of letters. I'm going to think of them as letters because I have a maths training. But if you're a chemist, you think of them as molecules, and if you're a biologist, who knows? And they go in a certain order, and then the helix is formed by having another chain actually going the other way, and the chemistry of DNA is such that they will bind to each other and form a helical shape, but they'll only bind to each other if the letters are paired up in appropriate ways. If an A is paired to a T and a G and a C, this is what Watson and Crick worked out in the 1950s, the information that it stores is in either one of the strands of the helix, but you don't need both for the information. Once you've got one, the other one is totally defined by the first one. So you can think of it as the information is in one or the other strands of DNA. Each link in the chain is the region of 4 angstroms. So is that 4 angstroms? I mean, unthinkably small. Unless you work in the kind of physics where you're always working in angstroms, and maybe you've got a mental image of what an angstrom is, but I don't. So the information is in one strand, and the information is essentially can be represented as a series of letters. And this is what genome sequencing projects do, is they get DNA samples from the genome of living organisms, and they work out what the sequence of letters is, and then you store those in a database, or you do whatever you study them. You look at how they change over time, and that's evolution. So that's what DNA is, and these are the watermarks that Craig Venter's team placed inside the genome of Cynthia. And they announced they'd done this. There was a press release, and that first date I gave you at the beginning was the date the whole project went public, and the paper was published, and they said, not only did we do that with all the genes we put in the organism, but we put these watermarks in. And Venter did a number of interviews, and dropped some hints about what the content of the watermarks was, but he didn't say what it was. So it was left as a puzzle out there. So, take a breath. First diversion. Who knows this book? No, not many, not enough. Everyone You should all read this book if you care about information and the way information can be passed and is held in living organisms and so on. There's many copies of it. It's still in print. I use this picture of a slightly beaten up paperback version because that's what my copy looks like. It's the kind of book, do you do this? I've owned about 4 copies in my life because you love it, and then you lend it to someone. You say, you've got to read this book, you've got to read this book, and they don't give it back to you because they love it as well. And so I'm on to my 4th copy of this. And the other 3 are all over the world. I don't know where they are anymore. When I did my PhD, so that's almost 30 years ago now, I guess, I essentially took 2 months off in my PhD to read. It's a fat book. To read the book and try and understand. It's not a textbook. It's if you're interested in information theory, it's quite readable. It has lots of funny stories in it. To try and understand what it was saying. And it really has changed my view of what information is, how information is stored conceptually, how it's stored, how it's passed on, copied, how can we try and hide that information by encoding it? Is there ever going to be a safe way of encoding information that no one can ever break the code of? And this kind of thing. It's an absolutely wonderful book. And I was very lucky. My supervisor kind of didn't say anything when I stopped doing any work for a couple of months. And it made a big difference. This is the description from Wikipedia, which is quite good. Where shall I stand to read it out? I like reading it out. The book discusses how through self-reference and formal rules, systems can acquire a meaning despite being made of meaningless elements, like the letters A, C, G, and T. It also discusses what it means to communicate, how knowledge can be represented and stored, the methods and limitations of symbolic representation. It does all that and more. It's a wonderful book. But of course, since I read that at the age of 24 or whatever it was, and then someone says, we've put these messages into DNA, but we're not going to tell you what it is, but we're going to drop some clues. What do? You obviously, many years on from your PhD, at this point I was an independent researcher, so I allowed myself some time off to go and play with this. I didn't do it straight away. I didn't do it the day it was released. But about 2 weeks later, I thought, Friday afternoon, I've got some time, not doing anything else. I will decode these messages. Wrote to myself a few scripts of Perl code to help me do it. Had some lucky guesses and got it mostly done on Friday afternoon. I took it home, sat there with my children, because you need one person on the computer and someone else kind of reading it out and saying, no, it's not right, it doesn't work. So I could do it with a 14-year-old who could read English well enough to help me. We finished it off that evening. And we found the messages in there. So for example, the whole thing is essentially a load of message, and I'll just pick out some bits and show you. So if anyone knows much about Craig Venter, they will not be surprised to know that his own name was encoded in there, as indeed also with the names of all the authors on the paper that had been published. And other elements, some of which we'll come to. But one of them said that, mail to MROQSTIZ at JCVI, which is the J. Craig Venter Institute.org. So, of course, I decoded the whole thing, but I found that bit by then. So, of course, obviously, you email them and you say, I've decoded your message, it's really cool. Well done, this is brilliant. So I sent them that email, and I got an email back from Michael Montague that says, Nick, you are the 35th person to decode. So I was quite pleased with that. I thought, it's not, I didn't start for 2 weeks, and I did it within a few hours, so, pretty good. The first was a recently graduated student from the University of Penn named Andrew Ettinger, 3 hours and 13 minutes after the watermark sequences were released. So he just I don't even know how he got it that quickly, but he just did it. So good for him. So I'd actually never been in touch with Andrew Ettinger until recently, and I thought I am going to try and find him, and he's not that easy to find, but I did find him. He works on the west coast of the USA now, and the picture's just off LinkedIn or something. And I had an email correspondence with him in the last week or so, and I said, "Are you the person who did this?" And he said, "Yes, I am." And I asked him some questions that I would otherwise have said something about in this presentation, but he hasn't replied yet. So I don't quite know what his story is. But anyway, he's the winner. I was number 35. So that was all fantastic fun at the time. I'm going to tell you a few more other messages. So those ones I've explained. There's some other stuff. There's all the other authors' names. And there were some quotations built in. So they encoded inside the genome of a living bacterium, what I cannot build, I cannot understand, from Richard Feynman, who I imagine everyone has heard of, which obviously is a very entertaining quotation to put in the genome of an organism you've just created. So that was a good one. I liked that one. And then this one, and this is by James Joyce. It's from his novel, A Portrait of the Artist as a Young Man. It's a kind of autobiographical story about someone in Dublin, Ireland, to live, to err, to fall, to triumph, to recreate life out of life. James Joyce, it said. Obviously, the reason they picked that is to recreate life out of life. It's very fitting if you're writing the genome of a living organism. But this is very amusing to me, that they picked some James Joyce. Here's a picture of James Joyce from the internet. Here's a book about James Joyce, which is actually the PhD thesis written up in book format of this gentleman on the right-hand side, as you look at it, who is my father. And my father did his PhD, and then his entire academic career, he specialized in the writing of James Joyce. And so I thought, oh, that's a really good coincidence. So I phoned my father up and I said, guess what? I don't suppose you've heard this story about synthetic bacteria genomes, because that's not the kind of news you follow, but I do, so let me explain it, because James Joyce is involved. So we enjoyed that very much. And he said, of course, you realize they're going to get in trouble for that. And I said, how do you get in trouble for writing James Joyce inside the genome of a synthetic bacterium? And he said, because James Joyce's work is all still in copyright. Right? And James Joyce's grandson, Stephen Joyce, very aggressively defends the estate of his grandfather, because there's money to be made, and doesn't let anyone publish anything. And anyone that's, of course, there's no one in the room. Anyone who has spent their life studying James Joyce, such as my father, has been threatened by lawyers multiple times. And if you reproduce a sentence of one of the James Joyce's books in your student handouts, and Stephen Joyce would hear of this, you would be threatened by lawyers to not do it. And there's been my father has had a couple of these letters that say you're not allowed to quote the work in that way without getting my permission and paying money. And it's a bit of a joke amongst Joyce scholars, like who got the most lawyers' letters. So, and indeed, my father was right, and this is from the webpage of a journal. This is the journal my father used to publish in regularly. It's the James Joyce Quarterly, now online. It wasn't online. We have physical copies of it lying around at home still. My father does. And this is the story they wrote about James Joyce and the first synthetic life form on their kind of blog page of the journal. And in there, if you could read it, it explains that how Craig Venter received a lawyer's letter from Joyce's grandson's lawyers saying, you can't write James Joyce quotations into a synthetic genome. I don't think the court case ever went any further. So that's a good story, but we're done. Act 1 is done. Act 2 happens between 2011 and 2013. And that comes about through the European Bioinformatics Institute. And on the top left is a picture of the European Bioinformatics Institute. And technically, my office is visible. Is it really? It's about there. My boss, Ewan Burney, is just above me. It's always sunny and bright like that. If anyone's been to Cambridge, you'll know it's always like that. And the bottom picture, so researchers live and work, work at the EBI, that's primarily my job, but the majority of the people that work there are providing data archiving and annotation and service provision for any molecular biological data from Europe and essentially from around the world. It depends who's in the room. For this audience, I'll say we are the foremost institute on Earth for managing large quantities of molecular biology data. If we've got any Americans, I'm sorry, and if we've got any Japanese in the room, you need to get your act together. And we have many of these graphs, and it doesn't matter, the details don't matter, but what you need to know is that the x-axis is time on a linear scale, and the y-axis is the amount of data the world expects us to store free of charge for them. and that's on a logarithmic scale, and almost everything is, vaguely linear on a logarithmic scale. So data is growing exponentially, and whichever field of molecular biology, if you know the subject, you're familiar with, the technological advances in the subject and the perceived importance of that subject mean that we are just generating data literally an exponentially increasing rate. And one of the problems this brings us at EBI is that our funders do not give us more money at an exponentially increasing rate. And so we have a headache of how we can continue to offer the service that the world expects on a kind of flat budget. I mean, we're not doing too badly, but not like that. And we had, a few years ago, a number of, starting in 2010, some sort of crisis meetings within EMBL. Around the same time, our American cousins at NCBI announced they were going to stop storing certain kinds of data. They couldn't afford to do it anymore. And we were worrying whether we would have to say the same soon. And if we were going to have to say the same, when would we say that? And should we just get on with it and admit it and save some money? And in the end, the crisis was kind of okay, and our funding went up a bit, and there were some good breakthroughs in the ways that we could compress the data, so it didn't get so bad. So we're okay. But it did mean we went through a period of really thinking very hard, sometimes in a bar in Hamburg, about how we would do this. And at the end of one particularly trying meeting, right when things were at the worst and we felt we were going to have to close some things down, we were sitting at the end of the day of this meeting in Hamburg saying, if there's any some other way we could store data, all the data is essentially stored on hard disk drives. And it's actually surprising how much of the vast amount of data other scientists do want to go back to. So the idea of writing it to a low-speed archive, like a tape archive, doesn't work very well for us. And so, isn't there some other way of doing it? And then we had a little, light bulb moment. And we realized that we had actually been working for all our careers with something that stores information for a long period at relatively low cost. And of course, that's the DNA in the genome. You know where I'm going with this. And of course, my research life before I got into storing information in DNA was genome evolution and ways we analyze DNA sequences or protein sequences to understand evolution. So I've been kind of staring at the answer. So this represents, the tree of all life ever on Earth. Humans are this little insignificant, one of these tiny little strands, tips of the tree is humans. This is all of life. And for 3.5 billion years or thereabouts, DNA has been transmitting information from cell to cell, from parent to child, from species to descendant species. Do that, and we thought, well, is this something we can use? And we didn't really think it would be very serious, but is there a way we can use DNA? We're not going to use living organisms. We take our inspiration from living organisms, but we don't want the ethics nightmare of manipulating the DNA of a living organism. But it's just a chemical. It's a chemical we're really good at handling in the laboratory. There's a lot of techniques. Can we use it as a way of storing information? So we realized, more or less all in one evening, that we pretty much had all the technology we needed to do it. There we go. So you need a writer to write the information, which for us would mean creating strands of DNA with a design that we chose. And actually that's really difficult, and humans have not got very good at doing that yet. But there are machines that can do it. The dominant technology at the moment, it came from a company called Agilent, which is a spin-out from Hewlett-Packard, who have a lot of skills in inkjet printers. And it's an inkjet printer technology, but instead of spraying dyes onto a piece of paper and allowing them to mix to make different colors, you spray chemicals which are modified versions of A, C, G, and T of DNA, and you do it sequentially so that you can build up a chain molecule to the design you want. So this is this is Agilent's machine, or it was a few years ago. There's other companies doing the same kind of thing now. And this actually, the other pictures are from a company called Twist Biosciences, who are taking the same kind of ideas and doing it smaller and with higher precision, so they can do more DNA in the same amount of space, and it's cheaper and faster. It's still slow and expensive, but it can be done. And this is just the kind of scale they do it on. I mean, the slide, they call it a chip. is kind of that size, and each one of those dots there is a little well, and actually in each one of those wells is 61 smaller wells, and in each one of those they can make a different design strand of DNA. And they can make a piece of DNA that's 250 bases or letters long. So that's currently pretty much where, and there's some errors, but it's not too bad. That's where the writing technology is. So the first time, making that first one is difficult. Once we've got the first one, humans are really good at making copies, and this is the PCR reaction for which a Nobel Prize was awarded, is this ability to bring into the laboratory a process very similar to the process going on in every living cell of any living organism, which is this process by which the two strands of DNA are separated, and each is used as a template to make a second copy of the other one. So you start from having one pair to having 2 pairs to 4 to 8. And very quickly, that's an exponential increase. And very quickly, you can make millions of copies. So once you've got the first piece of DNA, you can easily make millions of copies. So we have a writer, we have a copier if we need it, and we have a reader. Around 2012, 13, that was state of the art. That's from EMBL's laboratories in Heidelberg with my colleague Paul Bertoni. So one time in my life, I was allowed into a working lab. They didn't let me touch anything, which was a good idea. 5 or 6 years on from then, this is an Oxford Nanopore Minion device, is what they call it. It's just coming on sale these days, and it's way more powerful than that. It uses quite a different technology, and it's in your hand. In fact, in the last couple of months, they've announced the version of this which will plug into your mobile phone. So you have a DNA sequencer, could have a DNA sequencer with you at all times. I'm not sure quite why you'd want that, but I mean, checking your food is what it says it is. I can think of some more uses. So we have a reader that's reasonably, this one plugs into your laptop computer and can read DNA. And what we don't have at the moment, till the next slide comes up, is a language to put information that we want to store, that we might want to store, into the form of DNA. And that's the bit we did, really, that we invented in our project, is we created a code that would start with anything you could anyway store on your computer or your mobile phone. So basically, as we all know, that information is stored in a binary format in some way that we symbolically represent as zeros and ones. That's probably to do with magnetized iron oxide on a hard disk or whatever it is. So we said our input will be a file from a computer, or multiple files from computers, that are in the form of zeros and ones, and we had various stages in our process. But what we end up with is the designs for fragments of DNA that would store that information, which would allow us to recover it if we could reread the pieces of DNA, and that would allow us to recover if there were a reasonable number of errors. There'd be some error correction going on in there, and it would deal with the fact that the fragments of DNA we can make are not really very long. So we can't get all the information for one file from a computer into one fragment of DNA. So we had to have a system where we broke it up, and then you don't know what order they're going to come back in the reading machine, so then you have to have, each piece has to have a kind of index information in it to help you put it together again. So it's not really rocket science, but it's a bit I did, so I'm proud of that. So now we have a language, so now we can do the whole project. And so we thought, well, what as a proof of principle, what shall we encode in DNA and get it back? And back of my mind, I'm thinking I don't want to get a letter from any lawyers about things you're not allowed to copy and put in DNA. So I took the photograph of the EBI myself, and I thought, right, no copyright on that. That one's okay. And I thought, and we were trying to think, what kind of information. We'd seen the possibility that it might be cheap. In the future, it might be cheap, and it might be long-lasting. We know DNA can be very long-lasting. But if it's expensive to start with, the things you would store would be things of very high value. So what information is there that's very high value that the world doesn't want to lose? So we thought of William Shakespeare's literature, perhaps, would be a good example. We wanted to do all Shakespeare's plays, but that's too much. We couldn't afford that much synthetic DNA. So we settled on the sonnets, all sonnets. that William Shakespeare wrote, and they're available without copyright, because obviously he's been dead for many hundred years, and you can download them. There's a thing called the Project Gutenberg, which tries to collect the text of lots of classic literature as long as it's out of copyright and put it on the internet, and it's freely downloadable. So we got a file with all of Shakespeare's, I think, 154 sonnets he wrote. We got that from Project Gutenberg, so no copyright issues there. And we thought, the quote from Hofstadter's book, Gödel Bach, about the self-referentiality, or if there's any geeks in the audience, they'll know we love self-referential stuff. So we thought what we'll encode in the DNA will be Watson and Crick's paper describing the structure of DNA. So we contacted Nature and said we were doing this, and because it would be in copyright still. And they said, no, it's okay. So, there's a freely available version. You can have that. So it's okay, good. And so we had, written text from Shakespeare and a picture and a PDF document. In fact, it is. And even though they're all just binary, so really it makes no difference. But we thought it makes a nice example if we do different things. So we picked. I put a picture of Martin Luther King, but we picked. Martin Luther King's, the recording of Martin Luther King giving his I Have a Dream speech is on the internet and is available, and you can download it. And we couldn't do all of it because, again, sound files are quite big, but we took a little excerpt, about 20 seconds, from that speech and took that as an MP3, and we thought, well, we'll encode those because we love self-referential. Actually, I don't have a picture, but we had a fifth file we encoded as well, which was part of the description of the code we were using, because we like that kind of thing. So we put that in there as well. And we encoded all those in DNA, and then that was made for us by Agilent in California, and they sent that over to me in Cambridge. And in fact, I gave it to I'm useless in the laboratory, but I gave it to my colleague Paul Bertoni, whose picture you saw before, and he knows what he's doing in the laboratory. He did some of the preparation in Cambridge. He took it over to Heidelberg. He put it in the sequencing machine. The results get written onto a hard disk of what the components of the DNA was. It was brought back to Cambridge, and I decoded it, and everything was 100% right. So it's more than 6 million bits of information were perfectly recovered. And we wrote that up, and we got a Nature paper, so I was very pleased. It was my only first author Nature paper. So of course, we were delighted that had all gone really well. So, why would you do this, apart from it being a fun thing to allow yourself to do on a Friday afternoon? Actually, it took 2 years, start to end. But, then some of the reasons, and I'll say them again, maybe, just maybe, DNA will in future be a viable way for humans to choose to store information. And this just picks out some of the reasons, some of the features it has that are really good. So one is that, if you want to make 1 million copies of a bit of some information and distribute it, it's really easy with DNA. It takes half an hour in the laboratory, and you've got enough for 1 million copies. You just need to pipette it and send it to people. But it's much easier than photocopying a book, for example, or building 1 million hard disk drives. It lasts a really, really long time. And I know that because I've studied evolution, and as well as looking at the DNA of living organisms and thinking about what happened in the past, we actually have many examples where what's typically called ancient DNA has been found and recovered from samples, and it's been possible to read and understand the genome. And this is just one example. It's probably the most long-lived example, which is well accepted in the scientific community, and it's DNA from ancient horses. And they've got DNA from horse species of ancient horse that were alive 700,000 years ago, where they can get a DNA sample out and read the DNA and recover the information and work out pretty much the contents of that genome. And that's not a carefully designed sample of DNA. That's just a horse that died somewhere very cold. And what you really need for DNA to last a long time, the most important thing is that it's kept dry. And the easiest way to keep it dry is to keep it below 0 degrees. And then if there is any moisture around, it's frozen, it's not reactive. And of course, we all have the machine that lets us keep things near or below 0. So if you would want to keep that information in your own home, you just put it in the refrigerator. You don't need to worry about the DNA decaying. You need to worry about whether your refrigerator breaks down or there's a power cut. But we all have problems. And we'll always be reading DNA, unlike a floppy disk drive or punched paper tape, if anyone remembers those. Of course, this audience is too young and has no idea what I'm talking about. Think of all those ancient computer storage technologies that, the medium doesn't last more than a few years, and even if it did, we haven't got a machine that can read it any longer. DNA will last hundreds of thousands of years, and although we keep changing the machine we use to read it, we will always have a machine that reads it as long as we have technologically advanced humans who want to understand about their own health and their own bodily makeup and so on. So there's a few of the reasons. I'm not sure I was entirely convinced, but these are companies who have contacted us since 2013 and want to know about this kind of technology. We've spoken to all of these companies. I'm sure other scientists have too. I know some of them are actively working on this. Google, I think not. SanDisk, I don't know. IBM told us they weren't, but the day before that, someone had told us he was the head of their team working on it. So I think they're doing it secretly, but not very well. Huawei, don't know, but of all the people I've spoken to, they had the best informed scientists who knew what they were talking about. So I think they're doing something. Intel, I think not. Technicolor have been involved. Microsoft have invested tens of millions of dollars in this in Seattle and have a collaboration, very successful, going with the University of Washington, and are looking at this as one of their two most likely future technologies for information storage. So it turns out, maybe it really will work. Other people think so too. For me, of course, it was the coolest project of my whole career, and after 20 years of working on molecular evolution, no one wants to know. No one cares, but you do something cool with DNA and computers and stuff, and everyone wants to know. So this is my favorite slide. This is some of my various TV opportunities. I, of course, update it with the most important meeting I ever went to, which is over there, and some of the journalism we were featured in. So for me, it's been fantastic and a great opportunity to visit places I've never visited before. So I told my father, I've done this really cool project, and you remember that thing with James Joyce in some DNA? Well, I've done Shakespeare in some DNA. And of course, your parents are always really supportive and really pleased for you. So what did my father say? He said, you didn't use the Project Gutenberg Shakespeare, did you? And I said, yes, I did. I found it. I'm really proud of myself. And he said, everyone in academia knows that's the worst version of, it's full of errors. It's not been properly curated. So thanks, Dad. That's great. Thanks for the support. That's what you want your parents to say, isn't it? Excuse me. How am I doing? I'm running out of time. I'm only kind of halfway through, so I better speed up. Act 3, 21st of January 2015. So slightly before that, I received an email. You don't have to read the details. It's just remind me what to say. This is from the people that run the World Economic Forum meeting in Davos in Switzerland. And they invited me to go and present this at Davos. So it's really cool. I'll get to go to Davos and give a talk in front of all these bankers. Actually, if you're into it, it turns out, as well as all the finance and political stuff, they kind of do this sideline in scientific stuff, possible future of the science. I think it's meant to be entertainment for the bankers and the oligarchs. But it was really good. If you ever get a chance to go, I highly recommend it. They're very good at picking the scientists they get to do it, and they coach you on the presentation and making it accessible to non-specialists. And it was really good. So I was invited to go and speak at Davos. So of course I said yes, I would love to do that. So there's me speaking at Davos. And I gave a talk more about the research and why DNA was great for, and a bit more, slightly more technical detail than I've given you today, but not a lot more. And I thought, what am I going to do to make bankers and politicians interested in this? So I thought I'll concentrate on the fact that everything is digital these days. Everything we do is digital. Even money is digital. I thought I'll show them that money is digital, and I'll buy a Bitcoin, and I'll encode the Bitcoin in DNA. And then I'll tell them that's what I've done, and then I will give out the DNA, and if anyone can decode it, they can get a Bitcoin. So I thought it was quite cool. It didn't seem to go down terribly well at the time, probably because they were just all baffled, but I have scientists in the room, so I assume you're all with me. Up there, I never noticed at the time, if you look it's on YouTube, the presentation I gave is recorded, it's on YouTube, and if you scroll down below the line to all the comments, there's a whole bunch of nutters. out there, and there's a lot about intelligent design and God out there. And then there's one that says, is that Bill Gates on the right-hand side of the front row at 1 minute 34 seconds? So yeah, is that Bill Gates? Could be, couldn't it? Don't know. I didn't know if it was him. I didn't notice at the time. Yeah, so I did that with the Bitcoin. I didn't know much about Bitcoin, but I learned a bit about Bitcoin. And the point about Bitcoin is that your money, if it has a value, which is up to the community to decide, essentially exists in knowing a really long, complex, random-looking number that you don't tell anyone about. But the fact that you can put that through a function that provides a number, and you can tell people about that. So you tell people about the public part of it, and everyone can see the Bitcoin, but only the person that knows, or at least has access to, the bigger, more complicated number actually is the owner of it. So what you can do is you can buy a Bitcoin, you can expose the supposedly private number. Or I didn't expose it, we encoded it in a file in DNA. That's what the file kind of top left or across the top is. We wrote a file on a computer that said, well done on winning the Bitcoin, here's the secret number, here's how you go about claiming it if you've decoded it. And what I could do was publicize the public part of it, so anyone can look and see if the Bitcoin is, in a sense, still there. So I can show you my Bitcoin. Actually, what I'm showing you here is a different one, and it's 0.1 Bitcoins in the green box there. And so I can show you that, and you can't steal it from me. That's just me showing you my money. But if you saw this version of it, it's gone now, so don't try and steal it. Then you could take ownership of it. So that's what we put in the DNA to give away to the bankers. And that's what this little card is about, with a little test tube stuck, and I was giving these out at the end of the talk to Bill Gates and whoever was there. And we said, 3 years' time you can take the money, and if you haven't got it by then, I guess, I can't, this is public money, EMBL is publicly funded. I persuaded my head of outreach to pay for this stunt, but she said, well, if it doesn't get claimed, we'll have it back. So I said, okay, fine. Deadline. But because I can't bear to not do something geeky with it, I thought, as well as that, I will also stick in the picture of James Joyce. So it's not quotation, so I shouldn't get in trouble, but I just downloaded it off the internet somewhere. Google search, James Joyce, line drawing, because the file size is smaller. And I put that in. I told my father I'd done this, and I said, that's quite funny, I've just put this James Joyce. I think I'll get away with it. So that was the story about the Bitcoin. Nothing happened. I gave it out to Bill Gates and whoever, and nothing happened for quite a while. Act 4 of the story, December 2017 to 15th of January, which you'll note is just a few days before the deadline of the 3 years. And this is what happened to Bitcoin, starting on the left from when I bought it, a couple of $100, and over on the right-hand side is the deadline. This is what happens to Bitcoin over that time. Whoa! Suddenly, got up to $2,300 or $23,000 at some point. So it's suddenly become fun. So my head of outreach is saying, when does that end? When do we get the money back? She's starting to plan what she's going to do with all this money. And I'm thinking, yeah, okay, fair enough. That is what we said. So I thought, well, at least I'll give the scientists one more chance. So, we got on social media and I said, oh, it's almost 3 years. The competition's almost over. The Bitcoin's worth a lot more than it used to be now. And people would get in touch and they would say, is there still any of the DNA? Can we have the sample of DNA? Do you have any more? And we did have some in the lab. So I would typically write back to people saying, well, explain, why should I send you one? Because I could spend a lot of money sending these out if you're just going to put it on the shelf. So various people got in touch and they said, oh, I'm a molecular biology student, I'm working on this, and I think we can do it. And one of them was a guy I knew who's really hot on this decoding kind of thing as well. And I thought, he's going to get it. But all right, so we sent them out, something like that. It looks like that, in a padded envelope. Sent a few of the samples out, waited a few days. I'm working at home on a Monday morning. The deadline is Sunday at the end of the week. And I get this email, dear Professor Goldman. I'm not a professor, by the way. It's very nice. The students are always so polite. Dear Professor Goldman, with delight, I would like to let you know that, together with the help of some colleagues, I've managed to crack the Bitcoin challenge. I've claimed the Bitcoin yesterday around noon. You can find additional proof in attachment. In attachment, he sent some of the files that he had decoded from it. So, of course, what do? I run to the computer. I go to the browser. I look for my Bitcoin. And it's gone, there it is, minus 1.0002 bitcoins at 12:24 on the preceding day, exactly as Sander Voits, he's called Sander Voits, told me. This is Sander. He was a PhD student at the University of Antwerp in Belgium. He graduated earlier this year, and actually in this extraordinary world, small world of amazing coincidences, he's just been awarded a job at EMBL's Heidelberg laboratory. So he'll be working for EMBL from September this year. His PhD was in the fermentation of carrot juice. And in Belgium, you can get a lot of industry sponsorship to do a PhD in fermenting carrot juice. And there's a whole other coincidental story that I haven't got time to get close to telling you involving fermented carrot juice. So, of course, my first reaction was I was really annoyed that we'd lost all that money. My second reaction was, great, this is public relations gold. You know, how can we take advantage of this? I'm delighted to say that Sander got a fair amount of publicity. This is some of Sander's publicity. His favorite was being featured on Ukrainian television. Ukrainians sent reporters over to Antwerp to interview him. I haven't got the clip there. He mentioned doing it with his friends, and this is his friend Stein Rittek, who coincidentally I slightly know because he was a student on a course I'd taught earlier that year at EMBL, so I actually know Stein a little bit. But the real reason I like to show this picture is because now I can do the self-referential thing, because the first time I gave a talk like the one I'm giving today was in Antwerp to Sander's faculty. And rather than me explain the bit in the middle, I had Sander come and give this part of the talk, and he would explain how he did the decoding. So I get to show the slide of Sander giving the talk with the picture that I have in, but it's his picture, but I got him to give me the picture. So these things amuse the geeks. And one of the files Sander had sent me in attachment to prove he had done it was the picture of James Joyce I had put in there. And I said, do you know who that is? And he said, well, actually, I do know who that is, because when I did this, I went to my PR officer in the University of Antwerp and said, who's this? And he recognized it and said, oh, that's James Joyce, the famous, maybe not that famous, Irish author. And I said, oh, yes, it is. It's amazing that someone would know that, but you're absolutely right. And he said, yeah, apparently there's some people that work on James Joyce in the University of Antwerp. And I said, oh, really? So I phoned my father. And I said, Arnold, someone's decoded that thing I told you about. And not only did they get the picture of James Joyce, but they knew it was James Joyce. And they said that that was well known, or reasonably well known, at the University of Antwerp. And my father, at this point, then said, oh, yes, of course, I know them very well. There's a very strong James Joyce group working at the University of Antwerp. You should look them up on the internet. So I looked them up on the internet. And in another weird thing, the form of literary analysis they do at Antwerp and in other is called genetic analysis. So completely separately, they think this is genetics, but it's a different kind of genetics to my kind of genetics. And this is one of the things on their website. And then I was browsing around on their website, and the next thing I found was this page on their website with the same picture of James Joyce, which I then checked all over the internet for with Google Images, and that's where the picture is from. And I had originally got it from Google, who got it from the University of Antwerp. And it's by an artist who lives in Antwerp. And it's owned by the guy who studies James Joyce at Antwerp, who nearly came to do a PhD with my father, it turns out, but then turned it down because he got offered more money in Canada. So it's all a very small world. And my father said, actually, my father, who's been retired for about 15 years, on the phone then said, yeah, I was thinking I might go to the annual James Joyce meeting this year. It's in Antwerp. And I said, but Arnold, you haven't been for 20 years. You know, you've retired. And he said, yeah, I thought I might go. So in the end, he didn't go, but I did go. Because you do, right? So this is me, same shirt. I'm sorry, I should dress more excitingly. Talking to my kind of father's ex-colleagues, so to speak. I didn't stay for the whole meeting, but I went for the beginning and the dinner with them. And this is Sander and me and two more of the Joyce scholars who now work in Antwerp, posing for a photograph in the University of Antwerp. And I am just about done because apart from an amazing set of coincidences in the world, I really haven't got much more to say. And there isn't a big finish, except the usual corny, oh, everything's interconnected and isn't it amazing? But I mean, honestly, it's just amazing, I think. And I've not even told you the carrot fermentation story. But the bit, you know, at the beginning where I said who's a student and who's a PhD student, you know, I didn't, I kind of messed it up a little bit. I was meant to say, when Sander had emailed me asking for the sample, he said, I checked with my PhD supervisor. She says, yes, I can do it, and I can use the facilities in the lab, but not too long, just a few weeks, and then back to the PhD. And he was like down to the last few days, and he was going to give up for, and he did it. That was good. So my little plea is, if you're a PhD student or graduate student still, do that other ridiculous project that your supervisor doesn't think is part of what you're doing. Read the big fat information theory book, or take the time to decode something, or ask if you're allowed to do the ridiculous lab project that's going to get you $20,000 if you can do it. And if you're a supervisor of a student, encourage them to do that, or at least let them do it if they come and ask you for it, because really you wouldn't get science if we didn't do the fun stuff as well as the stuff we're paid for. And that's it, I'm done. Thank you very much for listening. And I'm sorry, it's a bit too long. I'm working on it.
It wasn't too long. I think everybody enjoyed it. Thank you so much for this really interesting and really enjoyable talk. I will start with one question. So when is the book coming? When is the book coming? Because obviously, so obviously the story would be, the competition for Dan Brown, for example.
Yeah, are there any ghostwriters in the room? I have no plans at the moment.
Okay, so do we have some questions?
Yeah, one question. You use for decoding 4 letters, of course, A, T, G, and C. But recently there were some papers that some scientists included 2 more letters, X and Y. Or 4 even.
They've got the one I saw was just like 2 weeks ago. Yeah.
No, I saw the one earlier. So yeah, would you consider using more letters? Would it be beneficial, or yeah?
So it would. So I'm not sure which one you're talking about with the additional 2 letters. There was something published just a couple of weeks ago with where they weren't storing information, but they were looking at making polymers with 4 other kind of modified DNA bases to give a total of 8. You're absolutely right. If you have more choices of letter, there's more information conveyed per letter. So that would be advantageous to do that. We're a long way from this being, commercially working very well. And the benefit of having more letters is relatively small compared to how far we've got to go. So it doesn't make it work yet. The main disadvantage is DNA is everywhere. We're used to working with the standard DNA. We have ready access to all the chemicals we need. We have machines that can write DNA inexpensively. We've just got to control it a bit better to write the DNA we want. And the advantage of people always being interested, there will always be a reader for it in the future, doesn't necessarily work for the unnatural versions of the chemicals. So it is interesting, and I was at a meeting recently and a couple of people were thinking of going that direction with it. But personally, I'm more interested in kind of following up how we would do it using the naturally occurring nucleotides and sticking with that.
Thank you for your amazing review on this this gigantic theme. But I have a question. What if someone will take this gene with not so good Shakespeare poems and add it to the virus, like simple one, and it will give it to the human body, so we will have a lot of copies of bad Shakespeare?
Well, so I said we don't put our DNA anywhere near any living organism. The way we encoded the information, one of the things we did was to make sure that it was very unlikely that any reasonably large piece of the DNA could be biologically recognizable, meaningful. So even, our DNA doesn't go near any living organism, and even if you put it near a living organism, it shouldn't have any biological function. So I don't think it's a major problem. I'm not the biggest expert in these biological threats, but we're not breaking any of the laws of scientific ethics. So I think we're okay. But I mean, as a more general point, if the ability to write DNA becomes widespread around the world, there are lots of evil uses you could put that to, sure.
And what maximum volume of information you can achieve in one sequence?
Well, in our little fragments are sort of 200 nucleotides long, and we get about 4 bytes of information in that length. You could get maybe twice as much in by doing a cleverer encoding. We were very conservative because we didn't want there to be any errors in the proof of principle, and people who've been working on that in the last few years have kind of doubled the amount of information. If you went up to having 8 letters instead of 4, you would double it again, essentially. But it's really, I mean, it's quite efficient, but then there's errors in all the stages of the process, and it's making a code that can correct the errors that's costly.
Thank you for your talk. And I would like to ask, when we store information, we have, like, for example, hard disk drives or magnetic tapes or perforated cards. And my question is, what is the physical form of what is the drive of this DNA sample? Is it powder or liquid?
So DNA, if in itself is a kind of powder, but you only use very small quantities. So I was telling a story, when the first sample was sent to me in Cambridge, it comes in these little plastic test tubes, these Eppendorfs, they're like 2 joints of your finger, that size. And I thought it was empty. And I thought something had gone wrong. And I took it to my colleague, Paul Bertoni, and I said, Paul, they sent it, there's nothing there. And he taught me what they teach them in the lab, which is, you hold it up to the light, and you look at the bottom, and you can just see a kind of film on the surface of the test tube. And it looks like that. If you actually filled the thing with DNA, that much would be a white powder, and that would be 2 petabytes of information, approximately. That terabyte is one of those disk drives that's like a small book. So 1,000 of those is a petabyte. So 2,000 is 2 petabytes. So it looks like a powder. Most people, more commonly in a laboratory, you'll see it in solution. So there'll be a small amount of liquid, and the DNA is in solution in the liquid. So then it's in a kind of, the DNA is still a chemical molecule, but it's in liquid. And that's very convenient for handling it in the laboratory and splitting a sample up into subsamples and so on. If it was going to be a device for long-term data storage, you might dry it out, which is better for long-term preservation, but then you'd need to do something else to read it again.
Okay, and as far as I know, when people work with DNA, they use special procedures. So to isolate the DNA coming from our skin, for example. And do you need like special procedures for reading and writing the information in your way?
No, so it's the other way. They use the special procedures so that they don't contaminate the sample they're studying with things from their skin. So it's not that it's dangerous, it's that you don't want to mess up the sample.
Not the dangerous part, but to ensure that you will be reading, for example, your information and not, for example, your genome accidentally.
So you might want to be, I imagine any device that ever got made would isolate the DNA, so it shouldn't be too much of a problem. It could be a problem if you had a lot of contamination, but if half of what you read is contamination, then it just won't decode into anything meaningful, and the other half will. So it doesn't destroy the information unless you overwhelm the important part with contaminant. So it hadn't occurred to me it would be a big problem. If you contaminated it enough, yes, it would be a problem.
Do you have any more questions? No? Lacey? Oh.
Slight comments. So, if I understand right, you put Martin Luther King's speech into your encoded DNA. Well, I have to inform you that Luther King's kids, grandkids have some similarities with Joyce's. As for Joyce, well, as he has died in 41, copyright has expired.
You're very well informed. So yes, that's correct. So James Joyce's work, very recent. The thing with the copyright, if that his estate was very clever with was if a new edition is published, even after death, copyright can also exist on that. So they actually kept the copyright going a little longer. So it would have ended in 2011, and in fact, I think they kept it till 2016. And it has just very recently, all Joyce is now out of copyright. And yes, I wasn't going to say, but you're right, there is some dispute about that Martin Luther King recording. And I didn't know about that at the time. And we've not had any trouble, but if that will be ironic if anyone ever does say anything.
Okay, so I would like you all to thank again Goldman for this amazing lecture.